Well, hi, I'm Ron Suskind, and welcome to the Autism panel here at the conference. This will be an interesting give and take. We have an extraordinary panel, and autism is something of a hot button issue. There are few maladies and few conditions that have drawn as much attention as autism in the past decade. There's some folks out there in public dialogue who compare it to some of the ways in which AIDS became the thing on everyone's tongue and everyone's mind decades ago. Polio before that. I think autism is different. We are dealing with probably a community of conditions. It's probably more accurately said that we are dealing with the autisms that are living underneath the umbrella of autistic spectrum disorders. Having said that, there is no doubt that if part of one's goal is to bring awareness to a particular condition, the way people are, what their challenges are, the last 10 years in autism have been years of success. I have spent decades writing nonfiction books, wrestling with presidents, dealing with issues of race and class, the complex issues of this era, terrorism, America's role in the world. And while I did that, there was something happening in my home, a story that now, of course, is the story out publicly of what was really happening in my life. Our son, Owen, 20 years ago, was a late onset autism kid, just shy of his third birthday. Kid who was chatting away, I love you, let's get ice cream, where are my Ninja Turtles, 250 words or so, 2.5-year-old speech, lost all speech. I had moved to Washington, D.C. to be the senior national affairs writer for the Wall Street Journal, where I spent that decade. Let's just say what was happening in our home was kind of all we thought about. Autism is a complex set of both conditions and challenges because it seems to be sort of the stuff of mystery. How a child who seems to be typical moving forward can seem to vanish in front of your eyes. It caused enormous concern for my wife and I. I first hear the word autism from a doctor a few months after Owen's speech, from a few hundred words goes down to a single word, juice. And I think a lot about that, about how we changed. When I first heard the word, my wife and I sitting with the doctor, it was like a blow struck us in the chest. Our son was sitting on the rug, playing with his hands, really lost to us at that point, and the doctor said, "This is called autism." I really didn't know much about it. I knew I watched Rain Man. Everyone saw Rain Man in 1988. First thing pops in my head, you're saying my son is like Dustin Hoffman in that movie? That can't be. And that's the last thing actually I heard from that doctor. Everything else was inaudible to both me and my wife. We actually sort of left our bodies at that moment. Now looking back, we remember like it happened last Tuesday. We're floating up near the drop ceiling, looking down on these two parents, saying, oh my, now what happens? Interestingly, we left those two people in that office. They didn't follow us out. I used to miss them, don't anymore. Because what was happening across 20 years is not only our son had changed, but we changed. We were different. We were learning from him and learning about ourselves, not in spite of, but because of the challenges that autism presented. Some of the parts that grew, the insights, are things that we consider some of our strongest muscles, our best selves. And I think everybody who deals with autism certainly deals with patients, the clinicians, folks who deal with the great army, a wave of people with autism, understand how this dynamic works. At the same time, it is a time of fear. Currently, 1 in 68 children in the United States are on the autism spectrum. 1 in every 42 boys. Now, some of these numbers may be fudgy, and there's a lot of debate about it, and we'll talk a little bit about these issues of incidence. But 1 in 42 boys, if you believe there's any credibility of that, is 2.5% of the male population. Epidemiologically, there's almost no precedent for an arc of growth. Now, is that because of widened diagnosis? Is it because we're looking at many things that we didn't know what to call now as autism or part of a spectrum of disorders? What's indisputable is that now we are moving to a point where this is not so much someone else's kid as a larger issue of public health. It costs about $60,000 a year to support an individual with autism as a child, $60,000 after taxes. Lots of things are still not covered, though 38 states now cover, as part of law, various treatments for autism. It is a life journey. Families are crying out for help. They're crying out for anything that's effective. There is some progress, and we'll talk about that today. There is not only extraordinary science that is starting to get its grip, rubber hitting road, especially in terms of genetics, but there are as well therapies that I think are growing and evolving. I think what's important to remember as we begin is that each of these individuals represents, in a way, a different set of areas of expertise and also orientation. Together, as we discuss what's happened, where we are now, and where we're going, I think that we will have colloquy, a virtuous set of exchanges. So let me start at the end with Ricardo Dolmetsch. Ricardo, why don't you just give us a 2-minute rundown of where you fit in the landscape of autism and what some of your extraordinary research is doing.
Sure. So I was a professor at Stanford for 14 or 15 years. I should just say I was, very happy doing what I was doing. I was sort of a basic biophysicist. I was interested in development, and then my son was diagnosed with autism, so I decided to change the direction of my lab. And eventually I helped establish a clinic to study autism spectrum disorders. And part of what we did over the course of, maybe almost a decade was to try and initially identify the genes that were associated with autism, and then subsequently try and develop cellular models based on stem cells. The idea being that it would allow us to get a biopsy of the brain of patients. And so that's what I did for all that time. And then a few years ago, I sort of decided to change direction because I realized that, well, actually, I had a call from my mom. And my mom, I'm Colombian. That means my mom calls me twice a week. And she gets expensive computer advice. And she said to me, you're making so much progress. And, every day you understand we know more about what causes autism. And it felt sort of like failure. So I decided that I was going to move to a place where I might be able to do something about it, and that's how I came to be at Novartis. And so I'm the global head of neuroscience at Novartis, where we work on many areas of neuroscience. But my passion and my interest is in these neurodevelopmental disorders because historically we thought that they were untreatable, and I don't think that.
I should also mention that Ricardo is the Colombian broad jump champion, I believe, and also represented them in the World Games. Ricardo is an extraordinary guy from an extraordinary family of brilliant eccentrics, and so he's just about the most interesting guy in the room. So let me just mention, just so you have a little idea of how each of these people is distinct. Chris McDougle, who I'll introduce next, of this panel, is really the Bigfoot, the expert in dealing with both research and clinical practice. Chris currently runs the Lurie Center of MGH. There are 3,000 clients there, 2,000 kids, 1,000 adults, or vice versa. And so what you get with Chris is someone who knows research, who's guided and shaped research, but also deals more than anyone else up here with the population, finding out what they're seeing, finding out what's working and what's not. So Chris McDougle.
Thank you. So I've been working in the field of autism for about 25 years, and I'm a child psychiatrist and an adult psychiatrist. And my initial interest in autism was in adults back in about 1988 when Rain Man was in the theaters. And I started a clinic at Yale for adults with autism, which at that time was really unheard of. Children with autism were barely heard of. And we got to know lots of individuals with autism from around the country. And I've continued my interest in adults. So when we came to Boston a few years ago to take the leadership of the Lurie Center, our goal was to see people across the age range. And we still do that. So from the time of early diagnosis until they're elderly. And there are very few centers that actually see people beyond the age of 22. So when you're 22 and you have autism, all the services end. You're out of school, and then you open the door the next morning and it's a black hole. There's nothing there. And those people are really only 25% through their life. So there's a lot of work to do in the trenches for people with autism to help establish meaningful adult lives for them. I've spent most of my career looking to develop medications to treat associated symptoms of autism. So things like aggression or self-injury or anxiety or hyperactivity or disturbed sleep. And this doesn't have anything to do with what we currently define as the core symptoms of autism, but if it can make a difference in someone's life if you can reduce self-injury and aggression and they're allowed to stay in school or remain in their parents' home instead of being institutionalized, or to continue to progress in a vocational area. So we're thinking about treatments for the core symptoms, the social and communication impairment and the repetitive behavior, but to date there hasn't been one drug that's been developed that's consistently been shown to affect any of those three areas. So that's one of our challenges.
Thank you. Mark Daly is the chief of analytical and translational genetics at MGH, at Mass General. And Mark is sitting on the sort of the wide arc of how we can use all the horsepower of computing, of understanding what we can understand in terms of gathering and interpreting data to really bring translation of the genome, translation of what we know genetically into clinical practice, into diagnosis, so rubber can hit road. People will feel effect in their lives. In Crohn's, obviously, you've done a great deal of work in that, but also in autism. So, Mark.
Thanks, Ron, and thanks for your powerful introduction to the session. I think it really sets a context for why all of us work on autism. When I first moved to MGH 10 years ago, I began working on the genetics of autism because, to me, as a scientist, I was committed my career to genetics because I felt this was the area of science and mathematics and computing where the greatest excitement was, but also the greatest potential for positive impact to the world was. So whereas, I think the greatest achievements in science, 100 years ago might have been in physics and chemistry, today they were clearly shifting to biology and most acutely to genetics, and the opportunity to bring genetics much closer to medicine was a powerful attractor. And the importance and the opportunity of genetics in autism was, to me, really the greatest biomedical challenge that any of us could take on as biomedical researchers, but geneticists in particular, because genetics, what's inherited and what's spontaneous in your genome that you were born with, is far and away the greatest epidemiologic predictor of risk for the development of autism. And so what that means simply doesn't mean it's going to be easy, but it means the clues to the biological underpinnings of autism that we ultimately need to understand if we want to have impact downstream, as Ricardo and Chris are aiming for, are contained in the genome if we learn how to read them. And so that's what our focus is and will continue to be for quite a long time, because this is not a short-term problem or a short-term investment that's needed.
Thank you. Last is Randy Carpenter. Randy's profile is as well unique on the panel because he's a doctor, he's a researcher, he does work in all of the issues of translational genetics, but also he is a guy who tries to make what we know actionable. He's a person who has experience starting companies and bringing some rubber to road. Randy?
Well, thank you, Ron. It's my pleasure to be part of this panel, especially when I consider that I'm the generalist up here and don't really have deep expertise like my colleagues do in any specific area. But my career has been through academics, large pharma, biotech has been focused on translational medicine. And for about the last 15 years, I've been leading small biotech companies that are focused on improving cognitive function. I think the reason I'm here today is the efforts that we had at Seaside Therapeutics, which really go back, the seed started about 15 years ago with a discovery around a mutation in a single gene, Fragile X syndrome, that dramatically, it's highly penetrant and dramatically increases your risk of autism. And we started studying these single gene, highly penetrant mutations with nearly Mendelian inheritance, with the concept that we could track the role of the protein that's not being made to a molecular pathophysiology, right, and then get targeted therapeutics that would ameliorate or correct the molecular perturbation that results from this genetic mutation. And with the concept that these could potentially be disease modifying. So rather than focusing on symptoms, focus on an underlying molecular cause, and with the promise that we could change the trajectory of brain development. And so that's really the hypothesis for the approach that we took. And while at Seaside Therapeutics, we raised a substantial amount of funding. We performed studies in 450 children and adolescents and adults with syndromic or idiopathic autism spectrum disorders. We guessed wrong on the primary endpoints in our phase 3 trial, which was fatal for our company. And it went out of business. But these experiences and the insights that we've gained have made me very optimistic that we're on the verge of developing drugs. And I'm in the process of hopefully starting another company that will have disease-modifying therapeutics that will improve the course of life for these children and their families.
Yeah, spoken like an entrepreneur. You can tell. He's the only one up here. Right? Yes, we can do it now.
I see opportunity.
We can take that next step. It's interesting you talk about Fragile X, because Fragile X was early. Fragile X was the first moment we said, oh, look at that. What I'm interested in is 10 years, 12 years, where we've come since those Fragile X days in terms of how much wider and maybe more hopeful, your kind of engagement, as well as Ricardo's, as well as Mark's especially, are now versus 10 years ago. And I'm going to ask everybody what you expected 10 years ago, what you have found now that most surprises you, and where is the area that you thought would work out better that didn't quite hit pay dirt. Why don't we start with you?
Well, when we started looking at Fragile X syndrome, we thought, this is really important for intellectual disability and how the brain doesn't really learn from experience as well of people with this genetic mutation. And we thought that it might be important for a small percentage of people, a pathway that we identified through studying Fragile X might be important for a small subset of people with autism, with idiopathic, autism from unknown causes. Okay. And I think that what surprised me from our clinical trial results is that a third to a half of the people in our trials with idiopathic autism had a robust response to treatments that were aimed at Fragile X. So I think one of the challenges going forward is, we have the presidential endorsement of precision medicine. Right. And the concept here is that the same drug is going to work in some people, but not others. Right. And it may even make some others worse.
Right.
Right? And we have to figure out why that is. Yet we still are in a drug development environment which is population-based, where you have to have an effect across a widely heterogeneous population, except for maybe oncology, where we celebrate 5% responders, reinvest, and then try to figure out why 95% didn't. There are many, many trials, I think, in psychiatry where we have, unexpected large results in 20% of the people. And those programs don't go forward. So I think that's what I'm optimistic that we're starting to be able to address some of that.
Mark, 10 years ago, you started into this genetic journey. I know it might be hard to think back 10 years ago, but what were you expecting then? And what have you found? The thing that's most surprised you positively, and the thing that you felt, hey, that was a bigger challenge than I would have thought.
Okay, so I think with respect to Fragile X in particular, I think what we didn't realize at the time was that it held additional clues to the underlying genetics of autism in a larger segment of the population. And we've come to recognize that this presentation of a gene with significant mutations that are highly penetrant for intellectual disability and partially penetrant for the traits of autism is now something that we've routinely come across in many other genes. And in fact, many of these are the actual targets of the Fragile X mental retardation protein. So there's a set of disorders that are quite like Fragile X in their presentation with respect to strong but very broad consequences neurodevelopmentally. This certainly not close to the majority of cases of autism. And in fact, the majority of cases of idiopathic autism have no obvious or recognizable mutation in their genomes. But it's a much broader category that I think opens up.
Define large majority. What percentage are in that large majority category?
It's probably about. It depends if you stratify. It depends on your diagnostic criteria. So
give me the good number and the bad number.
Okay, so the good number would be, the good or bad number would be when we diagnosed autism a generation ago, 75% of the time it was concomitant with significant intellectual disability. Today, that number is vastly smaller, so far less than half.
Or less.
Yeah, exactly. And so that isn't a change in genetics, and it's not even necessarily a change in environment. It's a change in diagnostics and what we call autism.
Right.
So in that severe group, a generation ago, it may be 50% of the cases, roughly, is what we estimate now, have a clearly recognizable and strong-acting mutation in their genome. Today, the number overall is probably closer to 25%.
Got it. Interesting. Chris, you've been at this for a long time. What's the longitudinal thinking for you? Back when you were a younger man in the mix, what were you thinking? Now, what have you learned?
Well, way back when I met the first person that I knew had autism, I thought this was going to be a relatively easy thing to figure out, to be honest, as a resident, because it seemed that the impairment was so prominent and that there was relative consistency across so many people that there had to be a unifying neurobiology. So the thing that I've learned over this amount of time is the real challenge of the heterogeneity of the clinical presentation and probably the genetic presentation, and that there may not be a lot of relationship between what we find genetically and what we see clinically. Right. So that really makes things quite challenging. So because idiopathic autism is so challenging, I'd say 10 years ago I was encouraging my junior people to go into things like Fragile X, because I thought they might be able to make more progress during their lifetime than others might. So, and that's sort of been a consistent theme. There are other well-known genetic syndromes Mark alludes to where
Was
there's
that good advice for those young researchers?
It was, but at this point, because of the challenges in getting federal funding,
Yeah.
it doesn't matter a whole lot.
Yeah. We can talk about that later.
Yeah, but I think as far as trying to do translational work with these guys to a clinical level, having something you know from the beginning makes it a bit more easy.
Yeah, yeah. Ricardo. I met Ricardo when I was reporting Life Animated, my book that came out in April. I went to see a guy named David Amaral, who runs the Mind Institute at UC Davis, who is really kind of a quarterback in the world of autism. UC Davis, the Mind Institute, is an extraordinary place. It's really kind of a mecca on the West Coast. And when I was originally writing this book about our 20-year struggle, our journey with our son, I thought I would do basic reporting. I'm an investigative reporter, so I thought I'd talk to all the leading experts. I did this book with my wife, who's also a journalist and much brighter than I am. And she said, don't put any of the science in here. Just write our story, the story of our family, and let them all debate it once the book is out. But in that process, I saw Ricardo in his lab at Stanford. That was about 2.5 years ago, where Ricardo was then and still is kind of the hot MB, the hot molecular biologist. You were there then, though, telling me about a guy named Shinya Yamanaka. And I'd like you to talk to people about the revolution of sorts, or at least the opportunity that it presented itself in 2006. Sure. When Yamanaka started to ask a different kind of question. Now he's won the Nobel Prize, of course, and what that did for your career.
Sure. So, when I started, when my son was diagnosed and I became interested in autism, the literature was bleak. It really was. It was bleak in the sense that neurodevelopmental disorders were, there were just not very many options. I mean, there still aren't, but the thought was that really they were not reversible, they were not treatable, there was nothing. And as I started thinking about it, there were a bunch of problems. One is we didn't really have any kind of molecular handle, but people were working on identifying the genes. People like Mark had started to do this. But the other problem that we had was that we had no way of studying the underlying, what it is that these mutations actually did. So, when you actually develop a treatment, you need something that you can study in the lab, and you need to be able to essentially iterate through a large number of molecules or antibodies or something. You need a good preclinical model, and there were no preclinical models. And so the problem is that, the preclinical models that we have were all based on mice or rats, and we diverged a really long time ago, and 65 million years have done, have made big changes in the brain. And so what we really needed was a kind of biopsy, a way of actually looking at the cells in the brain of somebody who had the disease. And this was not really viable until this Japanese group published what was a very surprising result, which was this idea that you could take skin cells or blood cells, convert them into stem cells, and then take those stem cells, and that was what they showed. And so they did this in mice, and so very quickly, many labs, including mine, started trying to see whether this could be done in humans, not because I was interested in stem cells at all. It was because I knew that we could start with stem cells and make them into neurons, because people had been working on this for a long, long time. And it suddenly opened this opportunity of generating a human model for a disease, a human cellular model, very much like they've done in cancer. And so the glimmer of hope, which I think now, 10 years later, is sort of starting to come to fruition, is that you were going to be able to develop a cellular model, and that you could use that cellular model to screen for molecules, to optimize molecules, and ultimately to come up with treatments. And I think, I have to say, at the time, I thought it was going to be much easier than it's turned out to be. I think now we have some projects. We have 2 or 3 projects at Novartis where I think we're making very good use of these lines, and they are a key part of the way in which we're actually running the project. But it took a really long time to develop the technology to the point where it was really robust and reproducible and industrial strength in the way that you really need if you're actually going to make a drug. So that's...
It's a beautiful rendition. Ricardo moves from the research community. Again, the Young Pioneer Awards, all the rest of those accolades. Now at Novartis, to do the difficult thing of creating a drug. And it sounds like some of the effort, the difficulty of that in the last just few years, you've just been there, has surprised you.
Well, yeah, it's, look, I don't have to tell most of the people here, making a medicine is really difficult. And making a drug for a neurodevelopmental disease in an environment where people think neuroscience is a bad idea, I mean, look, that it requires, Novartis has just redefined its core values. One of them is courage. And I keep telling people, you want some courage? You've got to help me out here. But the thing is this, look, we're in a disease area where there are no treatments.
Yeah.
So even small improvements are going to be revolutionary, right? Where, in fact, if there is one thing we can model in the lab, it is development. And we're not so good at modeling degeneration because people live a long time and, mice don't. But we can model development. We actually know some of the defects in these cells, and they're robust and they're reproducible, and we can come up with molecules, and we have. Yeah. The thing is, we have to solve that, then we have to solve all these other things along the way as well, right? We have to run a trial in children. So we just went into babies with a drug for spinal muscular atrophy, but that was a really difficult thing to do. There was no plan for this, right? So we're starting. So we have to, learn how to develop it. We also have to learn how to measure the outcomes. I mean, you should ask Randy how hard that is.
Yeah. And Chris.
Yeah.
It's a moving target.
But I think I expect that it's going to be as it is with most other areas of drug development. We will fail many times, but the idea is that when you fail, you learn from those failures. And I think that our approach has been to choose some of the easier things. Yeah. Where somebody has autism, but they also have something else, so that we can monitor the other things that they have as well. And we'll see if it works.
One of the things that's interesting is that if those who study the polio epidemic and what happened, they're surprised that NIH in those days was in the wrong direction for many years. It was the parent armies out there doing their surveying door to door, which started to produce data. I think they used the word data like we do then versus now, but that data started to reshape sensibilities. Jonas Salk is in the direction that many of these parent armies were pointing to. I like that story. I like that because it shows that especially when you've got something of such a wide arc as autism, where you do have armies of people out there, it is their life. It is every morning and every night. It's all they do and all they think about, is that we have parent armies. Frankly, many of the leading researchers and activists, clinicians, are parents. The profession is lousy with us. But what's interesting now is that we do have horsepower and opportunity we didn't have then with social media. It's made life so much easier than having to go out and find them and put them into categories. You can now post a Facebook page, and all of a sudden, they start sticking like to Velcro. And that's a very, very positive thing, because that's one of the areas of all of the miracles that technology was supposed to present and offer. It's one of the areas where I find actual utility that's actually changing people's lives. Those who are stressed, those who are in need, those who are dealing with a condition like this, have a place to go now where they can interface, possibly as subjects for research, with someone like Chris or Ricardo. And I think that's something that has changed fairly recently the lives of some of us here in the panel. Would you agree?
I mean, it's completely changed the way we recruit for a clinical trial.
Okay.
So, what are the low-hanging fruits in autism? Well, the low-hanging fruits are monogenic forms of the disease, right? They're still not all that low-hanging, because it turns out that even if you've got a single mutation, you end up with a very, diverse set of phenotypes.
And how many are we talking? 800? Is that the number?
Yeah, there are things that we're going into diseases that affect a few thousand.
Yeah.
But the good news is we know exactly what's wrong,
yeah.
right? And we think that there's some aspect of it that is druggable. We can work on. Historically, part of the reason why that would have been so hard is that just getting enough people to test your hypothesis would have been really difficult. And a clinical trial that takes 10 years to enroll is it's a non-starter. But now you can just have a Facebook page. Yeah. And it's amazing. Like, people come to you. And that makes all these things possible, right? So you're absolutely right and for them, of course, it is, and I can tell you as a member of one of these communities, it is just great to know what other parents that have kids with the same kind of mutation are going through, because it tells you sort of what to expect, right? What's the good news and what's the not so good news?
They can crowdsource each other.
Yeah, I kind of want to know,
Yeah. I mean, it gives you a context and it gives you a handle. I want to do a little fantasy camp here on issues of research. So we're sitting here 10 years from now. I won't have any hair then, but you've got time. Randy's already sold off several companies. Ricardo won the Nobel Prize. Chris will have, I think, some sort of cable show at that point for the world of autism. So what, 10 years from now, if I asked each of you, what worked? Either that surprised me, or maybe I'm thinking that this could be the thing. If I had to put money on a horse right now in the race, something that's happening in the research community that may have a fairly reasonable outcome. It's like predicting the election. I won't hold you to it. What right now would you pick that's giving you the most sense of possibility, opportunity, in terms of actual effects on this vast community? Why don't we start with you, Randy?
Well, what I'm hoping and what I anticipate is that I think we addressed it here. You can have the same mutation and have different symptoms, and you can have different mutations and have the same symptoms.
Yep.
But if you understand the molecular mechanism and you treat it, you should change function.
Yep.
And it should be meaningful. And it won't change in everybody, because that mutation may be on a permissive background or a resilient background, but it'll change a subset. And so I'm hoping that we will start looking at outcomes that are really important, that when they're improved, like in cancer, if you live, that's pretty important, right? And if you don't, that's not a good outcome, and it's pretty clear whether your drug worked or not. And I'd like to see us move to that versus a parent-reported symptom scale with big placebo responses and really noisy. And then you don't really know what the clinical meaning is. Do something that's clinically meaningful. Are the children, are they making friends? Are they developing social skills? Are they meeting their individual educational program goals? And we're going to have drugs. We have drugs that will do that, I think. And I think what Ricardo's talking about is looking at other consistent, objective phenotypes besides just the behavioral ones that are associated with these syndromic forms of autism. Right. I think that's going to be where we're going to look back and say, that's where we got a toehold.
Love it. Mark?
So, to me, I think the IPS technology, which is what Ricardo was describing, is far and away the most exciting thing to come available as a research tool right now, but potentially also eventually as a component of clinical practice. So in the short term, what it offers to us is the possibility of starting to make sense of the hundreds of genetic inputs which we'll inevitably get. And I think realistically now, as just exactly as Randy has said, we've recognized not only the complexity of the underlying genetics, but that in very, very few cases is it deterministic with respect to an absolute, certain outcome or trajectory in a patient given a mutation. I think we've seen this with, the 16p11 deletions and duplications, that the patients with those span the spectrum of autism completely.
But that's only about 1%?
That's about 1%, yeah, and so forth. But with almost every other of the newer genetic discoveries, this is also the case. There's a wide range of outcomes. So what if we're faced with, already, 30, 40 genes emerging from recent autism sequencing studies, 100 genes emerging from genetic studies of schizophrenia in the last few years, we already have, far more input than we know what to do with. And the potential of the technologies that Ricardo was describing is that we will be able to rapidly screen through genes and mutations, specific mutations seen in all of our patients, and understand the pathways that are disrupted, understand whether neuronal development is disrupted or altered in a certain way, understand if the expression of genes in a certain way is altered by these mutations. And what that gives us in the short term is an ability to understand at a higher level what the underlying biological pathways and processes that have gone wrong in autism are, bring that to the type of drug development that Randy was describing, and then in the future offers the possibility that new patients, not only with their genome information,
Right.
but with the information from those types of experiments done on their cells, might be able to be more effectively partitioned into therapies.
Right. Chris?
I think that in the next 10 years, we're going to be able to identify a couple of meaningful subtypes of autism that are currently considered idiopathic. We may not know the genetic underpinnings of them yet, but we'll better understand a pathophysiology. So one of the areas that's interesting to us and others is the role of inflammation in the body, and that there may be a subtype of autism and certainly other neurological disorders where inflammation, it may not cause the disorder, but it may contribute to symptom manifestation.
Right.
And inflammation is something that you can treat with a drug.
We know what to do with that.
What's that?
Inflammation is something we know what to do
We know
with.
how to do it, at least from a very global
Yeah.
perspective. And there may be biomarkers that we can use to identify patients with inflammation in their brain that may be amenable to treatment with an anti-inflammatory drug. So kind of an intermediate phase between what these guys are ultimately going to define and objectify and what we're doing currently as clinicians.
Yeah, yeah. I mean, the Brave New World, which everyone's talking a little bit about, is that I'm going to walk around with essentially my genetic map right here. And I'm going to get to this place of personalized medicine, which everyone talks about as kind of a Shangri-La up ahead, a kind of future of possibility, that the expressions of what we're finding genetically is going to help clinicians say, I know just who you are, and I know just what I think you need. Now, how is that something you might see, Ricardo, in your life, or will it be your grandson?
So I think the only way that's plausible, given our current financial models, is if it's combinatorial.
If it's what?
Combinatorial. If it's not, okay, so this is what I mean. So if I have to develop a drug for every single person, every single mutation, and there are 300, and I have to, the odds of that happening are very, very small, because there are going to be very few people with each one of those mutations. Many of them are going to be difficult to drug. It's going to be really tough. But I think there is the possibility that you might be able to cluster all of those mutations into nodes and pathways, and that you might be able to tailor combinations to people in a way that is a little less random from the way we do it now in psychiatry. So in psychiatry, in fact, that's what psychiatrists do, right? They do combine lots of things, but they just don't do it in a way that is very systematic. It's more like cooking, just a little serotonin here, a little bit GABA there. And if we could find a way in which we could do this predictably, and one possibility actually is to use people's stem cells. I'm not sure that stem cells will necessarily do it, but maybe some combination of stem cells plus genetics would give you some sense of what combinations to give somebody that are likely to be more functional. Now, so I think that's more likely to succeed than us making many, many drugs. I think it's just too difficult and too slow. Yeah. I think there are a couple of things that are happening now that are likely to be, that are likely to make at least the development of new medicines much easier, and perhaps even to be, much more, be therapeutically important in themselves. And that's kind of the development of wearable gadgets. Because I think that one of the problems that we have in general in neuroscience, in autism specifically, is that measuring the outcomes is very difficult. But in addition to this, there's just a whole universe of behaviors that you could imagine monitoring and then providing feedback on. And I think that there might be, we already know that education works, but we can't provide that level of intense education to most people.
Yeah, right.
Right? And one way of trying to do that might be to automate it.
Yeah.
And I think that is something that could work like, that's an engineering problem. I mean,
That is.
that could work, soon.
And a technology problem, but that's
Yeah,
doable.
That's a doable thing, and so I when I think about what am I going to do with my family, I mean I hope that something like that works. So that's one of the things. I have to tell you, inflammation I think is absolutely fascinating. I don't know what fraction of kids have of people with psychiatric disease in general have an inflammatory problem, but I'm becoming more and more convinced that there's an autoimmune aspect to it because that increase in the rate of autism smells a lot like the increase in the rate of autoimmune disease.
Yeah, right.
I mean, of course, there's the changes in diagnosis and all this other stuff, but there is a real increase under there, right? And and some of the genetics are also pointing to that, more in schizophrenia than in autism, but still.
It's interesting because one of the things that you hear from lots of families are saying, well, this sounds very promising, and but what do I do today?
Yeah.
I've been up for the last week with my kid. He's 6, he's 8, he has no speech. About 40%, 35% of the kids on the spectrum don't speak. That's a pretty sizable group. Many of them are using augmentative devices in this era, thanks to Steve Jobs. A surprise to Apple. And a delightful surprise. Wow, these iPads really can be just kind of a perfect middle ground that we can build all sorts of nice programs in them so that a kid without speech, often these kids have fairly strong cognition beneath their silence. And they come bursting out in one after another Helen Keller moment, water. It's quite moving, actually, to see it. It's extraordinary. And they are finally able to communicate, hitting symbols, and many type. When Owen, our son, was young, we had a leading psychiatrist, a guy named C.T. Gordon, who used to run NAR. His son was typing 150 words a minute with quite a bit of heft and quite a bit of acuity. I think in terms of the middle ground areas, which is what's going to probably bring impact soon, I just tell you, I stumbled into one because the twist in terms of what we did is that our son's passion, his obsession, so-called back then, was the Disney animated movies. Anyone here ever see The Little Mermaid? Let's see a show of hands here. Yeah, I'm so sorry for all of you. I mean, I've watched it 708 times. What we found early on is that that was his affinity. That's what we call it now. His perseverative passion. The view was, in terms of this sort of self-identifying phenotype, essentially, of autism, or these self-directed interests can be very narrow. They seem to be perseverative, obsessions. The view was cut them off or turn them into a behavioral tool to say, if you want to watch The Little Mermaid tonight, here's 10 things you've got to do, each walking across hot coals for you. We tried everything until we got to a point when he's about 9 or 10. We basically said, none of this is working. Cornelia, often it's the mom's acuity and heartfelt insight. She said, look, this passion is who he is. Love his passion means you're loving him. Let's just get in there. Let's just dance in front of the lit screen. So my son, as an example, there's many others, memorized all 50 Disney animated movies since Snow White was produced in 1937. I know the Nobel Committee is thinking of a family prize for all of us. We had to watch these movies incessantly. The more we did, the more we learned. The more we saw he was using them as a code breaker, essentially to crack the codes like the Enigma codes of the wider world. What's happening now that we happen to be at the center of is what's called affinity therapy. Almost every one of these kids has a specific one thing, several sometimes, but usually one first among equals. That is their thing. I ask them when I have roomfuls of kids, tell me what your thing is. Paleontology, astronomy, maps, train schedules, Disney, lots of Disney, Thomas the Tank, Minecraft. I mean, each one of them, they go 1,000 feet deep. And if you go down in there with them, you're like, my God, this is like an underground cavern. And because their need is so great, and they've turned these affinities into lifeboats, it's extraordinary the cognitive capacities that are expressed. Through Owen's Disney affinity, which he created an emotional language using dialogue and scripts of hundreds of hours, then he begins to draw. He learns because he watches the movies, and he learns to read by reading credits. Wants to know who was behind the screen of light. What's being tapped there? The neuroscientists who we're working with now at MIT and Yale say, oh, he's tapping the reward circuitry. If there are any neuroscientists in the room, they know that's the Gold Coast of what we're seeking here. Kids in the autism spectrum don't have a reward circuitry that works like you and me, or most of us, neurotypicals, whatever the devil that means. I mean, I don't know. I'm not that neurotypical. I mean, clearly Ricardo's not. I'm not sure about Mark.
None of us are.
You know, I think we're all part of a lot of spectrums that we're going to be able to see like bands of light over time. And these spectrums, just like the soundboard at a sound studio, have lots of dials that move across. And the question is how each of us can be identifiable as to strengths and weaknesses. What lots of the research that I find most interesting right now is finding, and some of them are neuroscientists that I kind of guide and deal with, is that what you've got with autism is a combination of hypo and hyper functionalities. All right? The hypo functionalities we measure exhaustively every 10 minutes, the things that the kid can't do. The hyper functionalities, which are largely born of what I call compensatory needs and strengths. The brain finding a way. We've talked about neuroplasticity. It's a word we use easily. It rolls right off the tongue. I don't think we're quite clear on what that actually impugns, what that actually means. Neuroplasticity is, in a way, a kind of revolutionary concept in terms of how much this thing gets shaped by the inputs, how fluid the brain is in finding a way, finding new neural pathways when stressed, when under siege. It's all just high school science, conservation of energy. It goes somewhere. And when parts of the brain are under stress, the question is, where does it go? What many researchers are finding in the middle ground, while they're waiting for the genetic miracles to occur, is that for every visible deficit, there may well be an equal and opposite strength somewhere, not if, but where. And so one of the things, just to throw out a little research that I'm involved in, is folks at MIT and Yale, Kevin Pelfrey at Yale, John Gabrielli at MIT, and others. What they're doing is they're bringing kids into an fMRI, and they're loading in whatever their affinity is, especially if it's visual, that helps. And finding, let's say it's Disney, when Beauty and the Beast plays, what neural substrates are being activated? Are they the most precious parts that we're looking for in terms of underlying capability that otherwise is invisible? And how then do we nourish those otherwise invisible strengths? Widen them. Bring in other sort of similar stimuli that are tapping motivation, intrinsic motivation. Because what I find now, as they come from every direction as I speak around the country, is everyone comes up after all the shows and say, here's my thing. I say, what did you do with it? Here's what I did with it. The interpretive capacity and some of the extraordinary innovations I find with kids for turning their passion into a world. Well, that's something the employers pay for. I mean, they get through the one-size-fits-all model of schooling, which we know is not all that fabulous for anybody. If they can get through that, or they don't get washed out into the discard pile because of that, you say, tell me what you're best at. Really? Yeah, tell me. Well, I'm not supposed to talk about it. Come on. And then we start to talk. And I'm like, well, that's something that's really extraordinary. What I hope for is someday we're going to have a set of metrics to measure these capacities so I can not only measure my son, who has a 74 measurable IQ, to put IQ scores on these kids. I think there should be a law against that. He does several things 4 and 5 times better than me. And I teach at Harvard. So which part of this you are going to keep? I think that is an area where everyone, both in the areas of molecular biology, in terms of clinicians like Chris, and the parent communities are saying, wait a second, we may be looking at the telescope in the wrong way. Let me just throw something out, which they said you should mention this because it's a beauty. Is that Tom Insel, head of the National Institute of Mental Health, was also part of the reporting. I saw him after I saw Ricardo. And I'm sitting with Insel in his office, and he's getting ready to give a speech. And he says, well, Suskind, you do speeches all the time. Can I try this out on you? So he gets the whiteboard up in his office, and he draws ASD in the middle of a circle, autistic spectrum disorders. Then around it, he draws a big square. And he says, okay, my speech is, well, it's about the divided landscape of all autism. It's like the Balkans. You know, it's a nightmare. And me, at the head of NIMH, I've got to try to figure out a way to manage these competing and warring communities. So what he does is he says, the key is the 4 I's. And he draws an I in this corner, and he writes up here, injury. Someone did this to my kid. It's a vaccine, it's someone, and it's something, and I want reparations, I want blame fixed. In this corner, he writes illness. We can't say what it is. It's probably something flowing through the genome, something maybe hitting some environmental triggers. We're working on that. Down here, he writes identity. All the folks out there saying, look, I'm not in the broken toy department. I know I'm a little odd, and I know it's hard to get dates. But I'm really good at some things. If I could be appreciated for those things, well, maybe we can manage the rest of it, the parts that's so hard. And many of the new technologies coming out, I've got a company building some of these, is helping to boost, to support the areas of deficit so that those compensatory muscles, the thing I'm good at, can shine forth. Identity. Don't call me somebody afflicted by a Holocaust or an epidemic. I'm just like you, just different. Celebrate the difference. And differently abled is not just a PC term, it's my life. And over here, he writes inquiry. All the neuroscientists following that great Eric Kandel line, the guru of neuroscience, that to understand autism means we will ultimately understand the human brain. What everybody here knows is that autism gives us a kind of way to look at the brain, both in the hypo and hyper functionalities that help us understand every brain. And so they're there too. So a couple months later, the book came out, and I gave a speech at NIMH, and I talked to Insel before. I said, so how did your 4 I's speech go? And he's like, not so good. I said, really? Well, I was going to mention it in this. No. I said, what happened? Well, each of the I's was affronted that there were 3 other I's. I said, oh, God. But the fact is that we are at a place where, frankly, I think these warring communities are coming together. We'll talk about this in a minute, and I'm sorry I'm going on so long, but something interesting happened I just saw. Autism Speaks. Everybody knows it's kind of, the sun and the moon and autism in this year. 10-year anniversary just occurred. The fact is they start out, Bob and Suzanne Wright, talking about this as Holocaust, as epidemic. What do? Let's find the causation. Let's get at it. Look, I don't think anybody disputes that. But many people, especially in that community of identity, said, wait a minute, I'm not somebody who essentially is deep into pathology. I'm okay. Let me do what I can do. Just now at the 10-year anniversary, Suzanne and Bob Wright was supposed to be in the panel today, but he's sick. They've been on the Lighted Up Blue Month, the Autism Speaks month of awareness. They've been traveling all over the world. They met the Pope and everything under the sun. But in the speeches Suzanne Wright was giving, she was not only talking about let's get a causation of all the horsepower Autism Speaks has, but also let's look at the great array of talents in this community. That's a change. That's a change. Having covered politics for many years, and I would probably dispute a little bit on that rat brain thing 65 million years ago. Having covered politics, I think the rat brain is pretty active still. Yeah, I do, definitely do.
Yeah. Well, I mean, it's not true for everybody. Some people have evolved more than others, so we're good.
So back to our panel. I mean, my question is, right now, when you look at this community, are you seeing a path to shared purpose and coherence where it comes to federal support, where it comes to maybe a nice idea as kind of a neuroscience bank, just like the infrastructure bank, something I talk to people in Congress about, a society moving to say this is something for everybody as a society to engage in? If that is not happening now, what will be the trigger that brings that kind of unity and uniformity? Chris, what do you think? You're right in the fight.
Well, the first thing I thought of is it might be such a prominent societal concern. I like your points about identifying strengths. And when you were talking about that, I was thinking of the strengths that many of my patients have beyond a knowledge of a topic, but sometimes their memory can be phenomenal. Their ability to categorize things and organize things.
Pattern recognition, off the charts.
Yeah. And
Companies pay for that, by the way.
you know, many people in society see an adult with autism walking down the street who's nonverbal, and they think that he has nothing to contribute to the world, that he's intellectually disabled. And I don't know that we know that.
I agree.
At least for a number of them. So I think assuming competence is important, and if this can be used as an example of trying to look at people's strengths and weaknesses among all of ourselves, then I think it could be very useful.
Yeah. Anybody else with a closing bit of hope? Ricardo?
Well, you know, I was going to say that there is some sort of movement in that direction, in the sense that there are now, I know in Sweden and now in Switzerland as well, there are foundation/companies being set up with the specific goal of finding things that people with autism are really good at, and managing all those other things that they have trouble with, like selling themselves, and you know, functioning in a sort of structured corporate environment. So, for example, I'm starting to work with one of these because I think that, you know, there are some things that you know, we could outsource to a group like that. I mean, I think you're right. You know, in fact, you know, I was a faculty member for a long time, so I had plenty of relatively autistic colleagues. You know, when I tell people, you know, what my goal is for my kid, I said, I just want him to fit in faculty meeting. You know, and, you know, but because, you know, really, you only have to be really good at one thing. The problem is that, you know, and of course, whenever I tell people that, you know, we are working on, you know, some drugs for some kinds of autism, I invariably get some hate mail from people who say, you know, you don't treat me. There's nothing wrong with me. You're and the thing is, of course, that, you know, yeah, there are some areas that people are very strong in. My observation is that, you know, sometimes there are all these other areas that just stop those things from showing through. And the question is, you know, what can you actually do to support people in, you know, executive function and attention and all these other things that make it difficult for them to function, which is one of the reasons why I think that perhaps this kind of electronics revolution is going to be important while we develop some new treatments, because I think that, you know, that is something you can do kind of quickly.
Mark?
So, actually, one of the things I've been really excited and encouraged by that's, you know, very much synergistic with what you were just saying is that while, you know, if you read genetics literature, it focuses always on the most severely impaired, syndromically affected individuals who have a clear and very significant neurodevelopmental disorder that has a single gene basis. That doesn't actually characterize the majority of patients with autism, kids or adults. And I think there's actually some really intriguing, you know, new genetic data that actually suggests that some of the genetic risk for autism actually is positively correlated with higher cognitive function in the quote-unquote neurotypical population. And there's very good reason to think that, you know, this line of thinking that we need to figure out the strengths and help enhance them is something that's going to be very, very effective in thinking about how we develop therapies for the majority of kids and adults with autism.
Let me go to our CEO, Randy. What do you see or think when you hear all of this?
I'm actually very encouraged by the changes that I've seen in the 15 years I've been working in the field. I think that when we started, there was very little NIH funding. There was very little appreciation. I don't go back as far as Chris, but many parents would go in and they would say, take care of yourself. There's nothing we can do for you. And there was this broken toy idea that something was broken, couldn't be fixed, there's no hope, learn to live with it. And I think what's happened is we have a Boston Autism Consortium that gets people to interact with each other. We have Autism Speaks that's come out since then that's created dialogue. There's greatly more NIH funding. We have the Simons Foundation.
Sure.
We have a fair amount of really smart people. We have really smart people like Mark and Ricardo. brilliant scientists working in the field, and it's attracting really good people with smart people. And this is a big problem. It's going to take a lot of really smart people, and it's going to take people working together. And I see that happening more and more.
Yeah, yeah.
And it's part of, I think, big data. People have to work together. When we started, you had my 200 patients that I've collected DNA on, I'm going to solve autism with it. And then pretty soon you realize you got to pool them with, everybody in the world to try to get some insight. And so it's kind of really, there's this really sense of collaboration that I think is wonderful. And I'm really, makes me feel optimistic.
Let me just finish by waxing a little literary. It's sort of my job, is I love what Chris just said about enlightened self-interest. I think I'm hearing that across the panel, is that we're seeing it through a new lens. I mean, I think Proust has a great line that the voyage of discovery is not about seeking new landscapes, it's about having new eyes. And I think what we're finding is that we are discovering new eyes. And eyes that tap the only thing that really creates any progress in this wide world, as the founders of this country knew, which is enlightened self-interest. Is that people saying, well, wait a second, there's something in that for me, and there may be a greater good that I can be a part of. That activates people. And I think part of what we're all feeling is that some of the understandable and crushing hopelessness that has abided and defined this planet of autism for the last several decades is lifting as we get new eyes. I'll just finish by saying I gave a speech in Tel Aviv recently. They have a big brain conference there just about a month ago. And before I did this in my public life, before I revealed my real life as a father of an autistic young man, I reported on the war on terror. So I know people in Israeli intelligence. One of them came up to me at the conference. Let's just say his name's Avi. And he says, hey, I love the last book. I said, the last book? The 1% Doctrine? No, the last one on your son. I said, really? Oh, yeah. We have a whole bunch of them, autistic young adults. We can't live without them in Israeli intelligence. I said, really? They see patterns in the data that are invisible to anyone else. They're our secret weapon. I love that. That sounds like a future to me. Thank you all for coming out today.